Introduction of Pharmacology. Follow BSMLT students. Pharmacology is the study of drugs including their origins, history, uses, and properties. The word pharmacology comes from the Greek words pharmakos, meaning medicine or drug, and logos, meaning study. Drug therapy plays a major role in the treatment of patients. It involves the use of drugs to prevent, diagnose or cure disease processes or to relieve signs and symptoms without curing the underlying disease. Definitions. Pharmacology is the scientific study of the effects of drugs and chemicals on living organisms, where a drug can be broadly defined as any chemical substance, natural or synthetic, which affects a biological system. Pharmacology is the science that deals with the study of drugs and their interactions with the living system. Pharmacology is the science that deals with drugs, their sources, nature and properties. Sources of drugs. Drugs are substances that are used or intended to be used in the diagnosis, prevention, treatment or cure of diseases. The major sources of drugs can be grouped into the following. Plant sources. Plant source for drugs are the leaf and other parts of plants, for example, barks, fruits, roots, stem, wood, seeds, blossoms, bulb etc. Plant part drugs. Leaves. Digoxin, digitoxin, from Digitalis purpura, foxglove plant, atropine, from Atropa belladonna, flowers. Vincristine, vinblastine, from Vincerosi, fruits. Physostigmine, from Physostigma venenosum, calabash bean, seeds. Strychnine, from Nux vomica, Physostigmine. Roots, from Physostigma venenosum, calabash bean, emetine, from Cephalozype cacuna, reserpine, from Rauwolfus serpentina, bark. Quinine, from cinchona, atropine, from Atropa belladonna, stem. Tabocurarine, from Chandrodendron tomentosum. 2. Animal sources. Medicinal substances are derived from the animal's body secretions, fluid or glands. Insulin, heparin, adrenaline, thyroxine, cod liver oil, musk, beeswax, enzymes, and antitoxins. Sera are some examples of drugs obtained from animal sources. 3. Microbial sources. Several life-saving drugs have been historically derived from microorganisms. Examples include penicillin produced by Penicillium chrysogenum, streptomycin from Streptomyces griseus, chloramphenicol from Streptomyces venezuelae, neomycin from Streptomyces fradii, bacitracin from Bacillus subtilis etc. 4. Marine source. Bioactive compounds from marine flora and fauna have extensive past and present use in the prevention, treatment or cure of many diseases. Coral, sponges, fish, and marine microorganisms produce biologically potent chemicals with interesting anti-inflammatory, antiviral, and anti-cancer activity. 5. Mineral sources. Minerals, both metallic and non-metallic minerals, have been used as drugs since ancient times. Examples include ferrous sulfate in iron deficiency anemia, magnesium sulfate as purgative, magnesium trisilicate, aluminum hydroxide and sodium bicarbonate as antacids for hyperacidity and peptic ulcer, zinc oxide ointment as skin protectant, in wounds and eczema, gold salts, solganol, or anophin, as anti-inflammatory and in rheumatoid arthritis, selenium as anti-dandruff. 6. Synthetic. Chemical derivative. A synthetic drug is produced using chemical synthesis, which rearranges chemical derivatives to form a new compound. Examples include acetylsalicylic acid, aspirin or ASA, oral antidiabetics, antihistamines, thiazide diuretics, chloroquine, chlorpromazine, general and local anesthetics, paracetamol, phenytoin etc. 7. Semi-synthetic sources. Semi-synthetic drugs are neither completely natural nor completely synthetic. They are a hybrid and are generally made by chemically modifying substances that are available from natural source to improve its potency, efficacy and or reduce side effects. Examples of semi-synthetic medicine include heroin from morphine, bromoscopolamine from scopolamine, hematropine from atropine, ampicillin from penicillin etc. 8. Biosynthetic sources, genetically engineered drugs. This is relatively a new field which is being developed by mixing discoveries from molecular biology, recombinant DNA technology, DNA alteration, gene splicing, immunology, and immune pharmacology. Drugs developed using living organisms with the help of biotechnology or genetic engineering are known as biologics. biopharmaceuticals, recombinant DNA-expressed products, bioengineered or genetically engineered drugs. Examples include recombinant hepatitis B vaccine, recombinant insulin and others. Pharmacological concepts, classification. Nurses learn to categorize meds with similar characteristics by their class. Medication classification indicates the effect of them on the body system, the symptom them relieves, or them's desired effect, for example oral hypoglycemics. Pharmacological concepts, classification. A medication may also be part of more than one class. Aspirin is an analgesic, antipyretic, anti-inflammatory, and antiplatelet. Pharmacological concepts, medication forms. Medications are available in a variety of forms and preparations. The form of them will determine its route of administration. Composition of them is designed to enhance its absorption and metabolism. Many meds are available in several forms. Medication forms: tablet, capsule, elixir, enteric coated suppository, suspension, transdermal patch. Pharmacologic principles. Pharmaceutics, pharmacokinetics, pharmacodynamics, pharmacotherapeutics, pharmacognosy. Pharmaceutics, the study of how various drug forms influence pharmacokinetic and pharmacodynamic activities. Pharmacokinetics, the study of what the body does to the drug, absorption, distribution, metabolism, excretion. Pharmacodynamics, the study of what the drug does to the body, the mechanism of drug actions in living tissues. Dose of formulated drug administration. Pharmaceutical phase, pharmacokinetic phase, disintegration of dosage form, dissolution of drug available for absorption, distribution, metabolism, excretion, drug available for action. Pharmacodynamic phase, drug receptor interaction effect. Pharmacotherapeutics, the use of drugs and the clinical indications for drugs to prevent and treat diseases. Pharmacognosy, the study of natural, plant and animal drug sources. Pharmacokinetics, absorption, the rate at which a drug leaves its site of administration, and the extent to which absorption occurs. Bioavailability, bioequivalent factors that affect absorption. Administration route of the drug, ability of them to dissolve food or fluids administered with the drug. Body surface area, status of the absorptive surface, rate of blood flow to the small intestine. Lipid solubility of them, status of GI motility. Next lecture, routes of administration. Thanks for watching. Follow MLT students.